everyone, we are talking about the DNA fingerprinting technique. The DNA fingerprinting basically involves simultaneous detection of markers in the genome that gives rise to a distinct pattern or unique pattern. So we had already discussed about the different types of markers and the criteria to choose a good marker. We will talk in detail about these markers and how then these DNA fingerprinting is basically applied for different purposes. So the first marker which we talk in detail is the restriction fragment length polymorphism. So what restriction fragment length polymorphism is basically, I explained you that the restriction enzymes, they cut the DNA inside and they produce different kinds of end. So what happens is that if we are comparing the DNA of two different individuals, that in one individual, for example, the restriction sites are present at these points, then in another individual, because of any reason that we talk of that give rise to the variations in the genome, one of this restriction enzyme site is actually disrupted. So there occur a nucleotide change, for example, or there occur an insertion or deletion here, so that there is that basically is disrupted. So this restriction, so what you will get is you will get different sized. For example, if you digest this DNA, you are going to get two fragments of say this X size and Y size. But if you cut this fragment of DNA, you are only going to get one DNA fragment which is larger than X and Y and for example of the size Z. So when you run it on the gel, then you are going to get distinct pattern, and this is basically length polymorphism, that for one restriction enzyme, when you digest two different DNAs, you are going to get different length. Poly means many, forms means different forms. Polymorphism basically means that there are many different forms that you will get based on the length, because it is length polymorphism. And when you are going to, for example, identify, then you are going to southern blot. So I said restriction digestion, running it on the agarose gel electrophoresis for visualization as well as creating a pattern, and then hybridization. So in hybridization, for example, if you use a probe, your gene of interest is somewhere lying here. It can be a disease causing gene, or it can be any gene that you are studying. If it is present somewhere here, and then you are going to get a different pattern for that gene. Because in this upper DNA, for example, if the gene is lying here, it is going to split into two and you are going to get two distinct bands. But in this, because the gene is not split, you are going to get one single band. So again, I repeat, marker detection and unique pattern. This is basically the DNA fingerprinting is all about. So this is the restriction fragment length polymorphism, again explained here in another diagram, which basically tells you that you create a probe. So when you do a probe which is encompassing this RFLP marker, then you are going to get distinct fragments on the DNA. So when you are doing DNA fingerprinting for an individual, this pattern would be very different from the another individual. Why? Because it may have variations, mutations, indels, changes, single nucleotide polymorphisms, etc. in its DNA, which is making it distinct from you, and therefore you are going to get a different pattern also, because some sites will be disrupted, some sites will be created, and you will get different lengths of the restriction fragments when you digest the two different DNA with the same enzymes. So this is restriction fragment length polymorphism. This tells you what is the VNTR. So I said the VNTRs is basically the occurrence of the variable number of tandem repeats. So this, for example, is tandem. Tandem I said is one after the another, and this is one block. So this block is occurring twice here. This is occurring, three times, not three times, basically this is another block, but you are having these two. Here again, you have got 2 blocks of this and 2 blocks of darker one. Here you are again having a different one. So you are having 2 here, 3 here, 4 here and 6 here, whatever. So when you are going to digest, for example, this DNA, and if you have got different digestive, for example, EcoR1. EcoR1 digest here and EcoR1 digest here. So if you digest this with EcoR1, you are going to get a fragment of this size. But in here, if you are going to get EcoR1 and EcoR1, it is going to be a larger size. Why? Because there is an addition of this one block here, addition of 2 blocks here. this will give you a largest fragment. So when you run it on the gel, the 4 patterns, the 4 fragments would be different. So that is how the VNTRs can also be used. Then there are what are termed as the microsatellites, which I said are the short tandem repeats. And in short tandem repeats, why does and how does it occur? It can basically, it is just showing the heterozygote and homozygote when they are present on the different alleles. So when they are present on the different alleles, again these are smaller, shorter, these are like 2 to 3 nucleotides. So here for example TAA is repeating, so TAA, 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 it is repeating a number of times. So they are different in a heterozygote, but in homozygote at one location it will be the same, but at the different allele position it would be the different one. So there will be a difference in that allele that can be recognized. One proposed application for the faster rate of evolution is the slippage during the DNA replication. So because of these things there can be some kind of slippage that can occur, and because of that they are basically evolutionary very fast in the adapting that is happening. So these are again the markers that can be used for DNA fingerprinting. So DNA fingerprinting, that is what basically the procedurally and principally is. I again review, procedurally it involves restriction digestion, it involves southern blotting, and it involves basically sometimes PCR as well, as we will see in our subsequent slides. But basically it requires this thing that you digest a DNA, you blot it onto this, and when the digestion pattern what you get based upon either the digestion pattern or based upon, we will see the amplification patterns, but based upon these markers you will get distinct pattern for all these different DNA from different individuals, and therefore you can use it for genotyping, you can use it for identity testing, and you can use it for a lot of different ways, and these different ways are what we are going to discuss now. So one major application of DNA fingerprinting is, for example, in forensics. So forensics is basically when you want to sometimes when there is a criminal investigation. So for example in this case, what they do is what is it basically is that because the DNA would be different and therefore you have the suspect and the victim DNA, you create a pattern. You generally use a number of markers, a number of different loci, not only rely on just one marker, but use a number of markers and identify the different patterns of DNA. And then when you compare that what is who would be the suspect in this case, and at the crime scene, DNA, because it is a very sturdy molecule, it can be isolated from any material which is, for example, blood, hair, any material. The DNA can be isolated, living organism ka jo material tha, and from that material, even from it is lying on the sputum or the semen, wherever that is, you can isolate the DNA and you can compare it with the different suspects' DNA, and you will see that you will have the identical pattern, and that can give rise to, that can finally tells us who the suspect would be. So this is one example of DNA fingerprinting that is given. As I said, the biological materials for DNA profiling, it can be collected from blood, hair, saliva, semen, body tissue cells, or DNA samples have been obtained from the vaginal cells transferred to the outside of a condom during this sexual intercourse. So basically, many a times it is going to ascertain the rape criminals, basically that rapists who have done. So these kind of material can also be collected and DNA can be isolated from them and used for the DNA fingerprinting as well. This is showing you an example, this is the final fingerprint, for example. So these are some 9 individuals, and 9 individuals you can see they might have probed with one particular region or gene, and you can see that for that DNA you are having distinct pattern for all these 9 individuals. There may be common bands, but if you look at the 2 lanes, then the pattern is quite distinct. In here, these 10 and 11 lane is basically the DNA of the zygotic twins. So as I said, the twins, the DNA is the same, and therefore you are getting the pattern the same. However, in these 9 individuals, it has been basically probed by the M13 phage DNA sequence, some sequence which is there, and you can see that the 9 individuals are having different patterns, and therefore you know they are distinct from each other. It can be used for identity typing as well. So this is how the results come in. This is another example which is telling you again about the forensics, how it is used for forensics. So you can see these are some of the samples that they have obtained, and these are some of the DNA they have isolated from the suspects, and they are isolated from the victim, and then they have run it on the DNA, and you can see that this pattern, this is from a vaginal swab, and this is basically similar to the suspect DNA or the DNA which is found here, and this is from different places where you get, and this is not the same. So you can see the identical patterns, and very clearly you can say that this is basically what is belonging to here. So this is another suspect, it is having a very distinct thing. This is the suspect which is basically having the same pattern as what was obtained from probably the vaginal thing. So this was a rape case, and it can distinctly say that the DNA is present there. Now there is another thing that when DNA fingerprinting was discovered, it was very revolutionary technique, particularly in forensics, and it was considered a very, very important evidence. But gradually people recognize that there could be errors. For example, in this case only, it can ascertain that probably this individual had an intercourse with the women, but it does not ascertain that she has been raped. So there are many other factors that needs to be considered, and therefore DNA, although DNA typing, DNA fingerprint is still a very important evidence, but it is one of the evidence which is used, and it is not the only evidence, because as we will see that it cannot ascertain, it can say the probability, but it cannot just ascertain 100%. Secondly, there are many lab difficulties also that occurs, because you need to have very pure DNA. If the DNA is very mixed, then the identification of the pattern can be ambiguous, and again it can give rise to some false things which is basically there. So some of the famous cases, for example, so this is the case, he was first, he was Colin Pitchfork. He was the first criminal which was caught based on the DNA. So fingerprinting evidence. He was arrested in 1986 for the rape and murder of 2 girls and was sentenced in 1988. So in this case, the first time DNA fingerprinting evidence was used, and because of this DNA fingerprinting evidence, he was caught and he was sentenced. So this was basically the thing. However, as I said, gradually a lot of lab difficulties was also identified. For example, the case of O.J. Simpson. So O.J. Simpson was cleared of a double murder charge in 1994, which relied heavily on the DNA evidence. However, this was in the criminal court he was relieved, but in the civil court, the family of the victims again filed the court, and he was found to be guilty, and later on he confessed to the crimes also, and he was given a 9-year imprisonment, and then he was released in 2017. So when initially the DNA evidence, and then they realized then there were a lot of difficulties in obtaining the pure DNA or identifying the unique pattern, etc., that they thought that he was not the double murderer, he was not responsible for that because of whatever evidences they got or whatever report they gave. So these are some of the cases, famous cases in forensics, and there are many more which is basically used for them. Apart from the criminal testing, it is very commonly used for paternity testing as well. So when the father is unknown, then again DNA profiling, because the basic principle is that a child obtains its DNA from the mother and the father. So when you will do the restriction, when you will do the DNA fingerprinting of the child's DNA, then you are going to get the pattern, which is some of the bands or pattern would be similar to the mother and some would be similar to the father. But there has to be some pattern which is similar to the father, and therefore some other would be unique to it, because there will be genetic recombination, etc. But there will be basically the pattern. It will not be identical to either mother, it will not be identical to either father, but it should have the pattern which is corresponding to the mother pattern and to the father pattern. So the paternity testing, so mother, generally they say mother is known because mother gives birth to a child. So mother, you can say with conformity that she is the mother of a child. But father then comes, therefore it is termed as the paternity testing, because generally you will have to find out, you will either the mother has to tell, or you have to find out that who the father is, and therefore the many cases arise for the paternity testing. For example, in this case, so this is a case, there is a mother, this is a hypothetical case shown here. So this is the pattern, for example, for mother, this is the pattern for the child, and these two are, for example, father 1 and father 2. So one of them is the father of the child. So if the mother is not going to truthfully tell, or even sometimes she doesn't even know who the father is, then if she is not able to tell, then what they do is they do DNA fingerprinting of the father's DNA. Again I tell you, but this is by consent. Generally you cannot obtain the DNA and do the fingerprinting. Any individual needs to give its consent for the samples to be collected, until and unless it's forensics that people are testing for the criminal cases. For the one has to volunteer to give its DNA for the testing thing. So if you compare now the pattern of the child with the mother, you can see there are bands which are corresponding to the mother pattern, and quite distinctively you can see that many of the bands of the child pattern is similar to the bands which is found in father 2, but not in father 1. Father 1 also there is one band which is identical, but the pattern is quite distinct. But with father 2, it is sharing many of the bands which is there. So many of the bands of mother, many of the bands of father, and some of the bands which are distinct to it also because of the variations. So you can clearly ascertain that the probability of father 2 to be the actual father of the child is much more than father 1. So this kind of paternity testing comes in, obviously, because this is probability. And then again, as I said, the DNA has to be pure, the DNA has to actually come from the father. It's not that we are saying that it is father 2, but it has come from somewhere else. So all these things we say that it points out that the father 2 is the father of the child. But again, there has to be many other reasons which are there, but paternity testing again can be done by DNA fingerprinting. So baby and mother are expected to share an allele, and the baby and father the other allele. So the two alleles in the baby, one is going to come from the mother and another is going to come from the father. If baby and father do not share common allele, the father is not the father. So not all the alleles, but basically most of the alleles, if it is not covering, basically fragments not covering, but allele it has to share, otherwise the father is not the father. If the baby and father do share a common allele, paternity is possible but not proven, because other men in the population also carry the allele at some frequency. So again, as I said, there is a probability that father 2 is the father, because if the population size is more, then we talk about the probability of arising of that particular allele. What is the frequency of that allele in a population? So frequency of that allele in a large population would be more. So there can be more person who are having a same allele, although they will not have. That is why I said that when you do DNA fingerprinting, you use multiple markers, you use multiple loci that is basically used to ascertain if every marker is showing that father 2 is the father by RFLP, by other markers, VNTRs, everything, then that is the father. But it is basically the probability that is told. Frequency of alleles that are shared in common by chance can be calculated, and an appropriate number of microsatellites analyzed to calculate probability of paternity. So now it terms as the probability of paternity, because we can do the Hardy-Weinberg law, we did it in genetics, and we can talk about the frequency of a particular allele which is shared with each other in the common places. To achieve high probability, 6 to 12 loci should be assayed. Exact number depends on variation in population for each marker. So as I said, multiple loci and then if each locus has few alleles, more loci are required. If allelic diversity is high, fewer loci can be analyzed. So allelic diversity needs to be considered, multiple loci needs to be considered, and therefore in fact sometimes multiple markers can also be considered so as to ascertain or increase the probability of paternity testing, and that is then it can be basically proved. So for example, I give you this chart. You can spend some 30 seconds on this chart and you can actually find out. So there are 3 couple patterns given here and there are 3 children, and what happens is at the time of birth some mixing occurred. So they do not know which baby is which couple's baby. So now you can spend some 20-30 seconds, or you can see it later. You can actually look at the patterns. For example, if you see the couple 1, then you can see the couple 1's pattern and you can compare it with baby 1, baby 2. Baby 2 has a very distinct pattern, and baby 3. So if you see, then couple 1's mother pattern is similar to baby 3 here. But this pattern is also, some of the bands which are coming is coming in baby 3. So probably couple 1's baby is baby 3, and similar way you can actually ascertain that what pattern it is giving rise to, or whatever baby 2 or baby 1, you can actually allot it to different couples which are here. So this pattern is here, and then this band is also coming here, this band is also coming here. So you can actually compare the different bands, and then you can say which baby is which baby, which couple's baby. That can also be done. Another application is the gene finding. So that is basically about the forensics in the criminal testing or the paternal testing. Another application is what is called as the gene finding. So using the basically screening for RFLP markers, for example, on a chromosome, we can approximately determine the location of a gene. How we can do that? We can find the difference between the RFLP markers of family. We can actually see the patterns in that, and we can then locate it to a particular loci or to a particular gene. Although when we see the distinct pattern, we can find the patterns, but it is difficult to know where the exact gene is located. So exactly the base pair to base pair, it is difficult to locate, but at least we can allot it onto the genomes, onto the chromosomes, that way to which chromosome does it belong to. Because whatever we are probing with, that gene of interest is what you will probe it with, and when you are going to probe particular chromosomes, then in some of the chromosomes, for example, it shows a pattern, but in some of the chromosomes, so then you can say that this is present on 6 chromosome, and in that 6 chromosome also, if it is showing at different fragments, you can create a map and you can actually locate it, that how these fragments are related. If you do the partial digestion, how these fragments are joined together, and then you can locate your particular gene as well. So this DNA fingerprinting can also be used for gene finding. DNA fingerprinting is also used for epidemiology. So if you want to, for example, predict a particular epidemic or spread of a disease, or for example, in this case, it is showing the comparison of DNA from the patient and the food. So what happens is that some of the individuals, they consumed a juice, and that juice was contaminated with E. coli. So if there was a contamination of E. coli, then obviously the DNA isolated from the juice that they drank, or the E. coli isolates from them, would give a similar kind of a pattern. So the people who do not consume the contaminated juice because they do not contain E. coli. So if you isolate the DNA, the E. coli isolates that you get from those individuals, you will get a different pattern. But if the people have consumed that contaminated E. coli containing juice, so you can see that you can actually, by DNA fingerprinting, by using a probe against the E. coli isolate, one of the gene or whatever pattern you want to have for the E. coli isolate from which is basically present in the juice, then you can actually do the patterning and you can see that the E. coli will be same. However, the people who have had juice which is not containing the E. coli isolate, then they will have a different pattern. So again, DNA fingerprinting can be used to predict this epidemiology. This is, for example, a contaminated food we are talking about, but this can be a diseased conditions as well, because in diseased conditions too, what will happen is the DNA pattern is going to different. If we are isolating certain isolates from them, if for example a particular virus, it resides the lungs, then if you take some sample from the lungs, for example, if it is in the lungs, some kind of a matrix there, or some cells, or some whatever in some water which is there, or some kind of sample that you are going to isolate it, and then what you are going to do is you are going to fingerprint it. So where there is no pathogen, that pattern is going to be distinct from, because the pathogenic DNA would also be there with the human DNA. And then you can say that these are the differences in the diseased and the non-diseased thing. Obviously, you will use the probe either for that pathogen, complete pathogenic genome, or if you want to talk about one particular gene of that pathogen, then also you can use the probe and you can have these different epidemiology that can be studied. So other applications of forensic sciences, that of DNA fingerprinting, we have talked about the biological evidence. So in biological evidence, the major thing is to do about the paternity and maternity testing, to talk about the linkage of the suspects to the crime scene. So this is basically we have talked about. For the diagnosis and development of the cures for inherited diseases. For example, DNA fingerprinting is a lot of times used for the diagnosis of cystic fibrosis, hemophilia, Huntington's disease, familial Alzheimer, sickle cell anemia, thalassemia, and many others, because these are all genetic inherited diseases. When you compare the children pattern with the parents pattern, and then you are going to see it in the lineage, and you identify or use the probes for the DNA differences that occur in these diseases, you can actually see the inheritance of the diseases as well. So they can diagnose, and they can also aim for the development of cures. For example, we can also see that in a particular disease, if the pattern is distinctive, we can actually identify those fragments and we can sequence them and see if there is a difference in the genes that is occurring between the two. So we can ascertain that in particular disease, this is the gene that is basically affected or not, and therefore we can use the treatments directed towards that gene and we can cure it. So that is also one of the application of the DNA fingerprinting. The education of prospective parents to do the risks of having an affected child. So identification of the DNA patterns, as I said, associated with the diseases to help establish the treatment. So they can be, the samples can be taken from like amniocentesis, some amniotic fluid can be taken, and the genetic disorders, they can also be detected by DNA fingerprinting because they will have a distinct pattern as compared to the normal child that is developing. Identification of the individuals. So sometimes DNA fingerprinting is also used for identifying the missing persons or the casualties. For example, if there is a bomb explosion or if there is a flood, so it is very difficult to identify the victims who are there. So then the DNA can be isolated from the casualties, from the servicemen if they are missing, or from the calamity victims. As I said, they can be fingerprinted compared to either the database or their relatives, and then the identity can be tested of the individuals which is there. Then veterinary applications, the parentage testing of the animals which is there, the wildlife studies, identification of inheritance pattern of genetic diseases in animals, inheritance of genetic patterns, identification in population, investigate genetic susceptibility of the population of diseases. So even in animals, not just in humans, the distinct pattern can be identified in all these cases by using the DNA fingerprinting. Not only in animals, even in agriculture, there is a lot of application of DNA fingerprinting, because if you have to breed the dairy animals, so for breeding of dairy animals, you can identify the distinct patterns of, for example, large volumes of milk producing cow. So, and then hybrids can be generated. So, even the patterns can be identified for the cultivation of various kinds of crops. So, agriculture, again, the various kinds of crops can be cultured based upon the different patterns that we obtained or hybrids, the patterns we can obtain, we can see that these are the highly producing crops, these are the low producing crops, so we can identify the distinct patterns, and again based upon those unique patterns the agricultural applications are there. So DNA fingerprinting is all about basically the unique markers, the markers, detection of markers, simultaneous detection of markers, and basically the identification of the unique patterns. That's all for today. Thank you.